Leaky gut for improving breeder health. So just an overview of what I'm going to talk about today, a little bit of background about why we're interested in the intestinal health of chickens, an overview of the anatomy, a little bit about the microbiota, which we've heard a bit about already, and the different factors that affect gut health and might influence leaky gut, and what we can do to try and reduce that. Okay, so why are we interested? Well, the gut's the largest organ in the body, and it's responsible for the breakdown of feed so that it can be absorbed and the nutrients can be absorbed. It's one of the major interfaces between the bird's immune system and the environment, which we've heard a little bit about today already. And if you have poor gut health, it can cost around 6.8 pence per bird, or 11 cents. And gut health has a pivotal role in the efficiency of production. But what is gut health? Well, it's the ability to be able to defend against enteric pathogen, the capacity to be able to break down the feed into its constituent parts so that those can be absorbed as nutrients for the birds, and also the ability to tolerate normal fluctuations in gut conditions. So gut health requires optimal tissue development, optimal immune system development, and acquisition and maintenance of a favourable gut microbiota. So a little bit about the anatomy of the gut, which probably most of you know all about already, but we have the beak, of course, and the crop. The crop is where the feed will be stored before it's transferred to the proventriculus, which is also known as the true stomach. This will then pass into the gizzard, where it's mechanically broken down, and this will pass then into the small intestine, where most of the chemical breakdown of the feed occurs, and the nutrients from this will be absorbed. And then the ceca and the colon, where we get water reabsorption, and in the ceca some fermentation as well. So essentially, the gut is a specialized tube that runs from the beak to the cloaca. It's divided into distinct regions, each with a specific role in digestion. And each region of the gut relies on correct functioning of its neighboring regions in order to function correctly. The last few days of incubation are really important for gut development. And an indicator of poor in-egg development is the poor gut tone early in life. So here I've got some images. We've got at the top the duodenum, in the middle the jejunum, and at the bottom the ileum. This is a picture of good gut. We've got the colour is good, it's a nice pink colour. The jejunum at the top, you can see where the intestine's been cut open, the gut has rolled back, and this is an indicator of good gut tone. As we go through the small intestine, you can see that the feed becomes darker and drier, so that's a good indication that good digestion is taking place. We've got electron micrographs on the sides here, and at the top we've got an image of the villi. So there's good distribution of the villi, and these are able to move freely. And then at the bottom we've got microvilli, so we've got a good coverage of microvilli that are evenly distributed. So gut development. Villi development begins before hatch, really. And in the first 4 to 10 days, the villi will reach approximately 50% of the adult villus height. And this is due to the rapidly dividing cells, which are highlighted in red here, which are both at the bottom and along the edges of the villi. So an adult villus will have mature cell turnover as well. You still have cell turnover even in an adult villus, but the cell turnover, the dividing cells, will be typically at the bottom. And any stress during brooding will have an impact on the development of these villi. So stressed birds have been found to have shorter villi, up to 19% shorter, and also lighter gut tissues, approximately 26% lower than healthy birds. So this really does have a major impact. The villi are covered in a single layer of cells, and this is a barrier between the bacteria entering the body. So any loss in intestinal integrity can result in bacteria crossing this barrier. So here I've got an image of the normal gut. In the gut lumen, we've got a collection of different species of bacteria, the gut barrier, which is a single layer of cells, and then beneath that, the gut tissues. And in a compromised gut, we can have gaps between these cells, which allows bacteria from the gut lumen to enter the gut tissues and potentially into the circulatory system. So if we have failure of the gut barrier, the gut can become inflamed, and this increases chances of disease. It also reduces nutrient absorption, which results in poor growth rate and can result in nutrient deficiencies. It can also result in opportunistic infections such as necrotic enteritis and femoral head necrosis. Also, bacterial toxins can cause toxic shock in multiple organs. Here I've got another electron micrograph of healthy gut tissue. So as you can see, the surface is quite smooth. And here I've got another image where the gut surface has been compromised. And here you can see the surface is no longer smooth, and there's damage that has occurred to the cells here. This is a zoomed in image on one of these villi. And here I'm not sure how well you can see, but we've got bacteria that are colonizing the surface here. So what does good digestion look like? Here I've got a couple of images of droppings. We have cecal dropping, which is quite wet looking, but this is normal. And a fecal dropping, which is drier and also has a uric acid cap. So these are normal droppings, and these are associated with a balanced gut microbiota. But what does disrupted gut function look like? So here I've got some images again. The furthest, okay. Won't touch that again, sorry. The image furthest away shows that there's still constituents in the feed there, that it hasn't been good digestion. The image in the middle is a shiny, kind of wet-looking feces. Again, this is an indication of poor digestion. So the shininess is fat content, which could indicate that fat digestion hasn't taken place effectively. And here we have some gassy ceca. So the bubbles in the ceca are indicative of different gases being present. And these are all associated with disruption of the gut microbiota. So what is the gut microbiota? Well, it's a large community of bacteria that live in the gut. The different species can vary depending on the part of the gut that you're looking at. They can change with the age of the bird, and also change with diet composition as well, which we heard earlier on. Trillions of bacteria, made up for between 700 and 800 different species, are a mix of both favourable and unfavourable bacteria. And this microbiota plays a major role in animals' health. So the microbiota promotes the development and maintenance of the gut tissues. It stimulates the development and maturation of the immune system. And this continuous stimulation by the gut flora ensures that the immune system is in a constant state of readiness. And the microbiota modulates the immune response. It also assists with digestion and provides beneficial metabolites to the host, and offers protection, or offers some protection from gut pathogens by means of competitive exclusion. So if all the receptor sites are blocked by beneficial bacteria, then potential pathogens are unable to find a receptor site to bind to. There's also competition for nutrients, so beneficial bacteria will compete with potential pathogenic strains of bacteria for nutrients in the gut lumen. There's also the production of inhibitory substances such as bacteriocins, which are natural antimicrobial compounds, and also organic acids, which lower the pH. And a lower pH is less favourable to certain species of bacteria. So the development of gut microbiota can be likened to the development of a forest, where we start with the bare ground, and this is colonised by moss, grasses and ferns, followed by small trees and shrubs, and eventually mature trees. And this happens over a period of time. So the gut at hatch can be likened to bare ground, and the mosses and grasses and ferns can be likened to pioneering species of bacteria. Small trees and shrubs, the succession species, and eventually the mature species, the trees. Any stress on the gut system can slow this rate of succession, and each time the gut is compromised, the gut flora is set back, and this requires some re-establishment. The microbiota of a chicken takes a few weeks to fully mature, and this maturation process can be disturbed, leading to dysbacteriosis and/or wet litter in this time. So if the development of the gut of a young bird is compromised, then also the maturation of the microbiota is impaired as well. Here I've got some pie charts that show the breakdown of the different genus of bacteria present both in the ileum and the ceca, and the good ileum compared with the poor ileum. So in the ileum, the major genus of bacteria is Lactobacillus, with some Staphylococcus E. coli and also some Clostridium species present. And these in the poor ileum will have more Staphylococcus, more E. coli, and more Clostridium compared with the good ileum. In the good ceca, we've got several different species, and in the poor ceca, we've got even more different species, and there's an overgrowth of certain bacteria present here. So shifts in microbiota are indicative of malabsorption. You can get poor fat absorption, poor protein and sugar absorption, and if you've got fat, sugar and protein all present in the ceca, there's more nutrients for bacteria to grow. Resulting in bacterial overgrowth, we can have production of different gases such as carbon dioxide, methane, and hydrogen sulfide. You can have toxic amines produced, and these will irritate the gut and result in growth depression. Bile acid inactivation will impair fat absorption, and we will get an immune response, which will result in a leaky gut. This then leads to further disruption, and an intervention may be required. The impact on the microbiota is usually the same, irrespective of the factor that causes the disruption. So next I'm going to talk about the different factors that affect gut health, and these include poor gut development, which could be in ovo or could be post-hatch, the feed, infection, water quality, and environmental stress. These factors can be synergistic and therefore can result in more severe intestinal upset when combined. Systemic disease challenge can cause immunosuppression and physiological stress, and enteric disease, both clinical and subclinical, can cause disruption to the gut tissues and microbiota. An enteric disease can be viral, bacterial, or might be parasites, or such as coccidiosis. So we've heard a bit about coccidiosis already, so I won't go into this too much, but coccidiosis is one of the major intestinal issues in poultry and should be controlled and monitored. We've got feed. The form of the feed, such as poor pellet quality or excess fines, can increase the rate of passage, and this will result in poor efficiency. The actual raw content of the feed will influence the bacterial species that are present in the gut, and any changes in nutrient density will alter the gut microbiota composition. Also, mycotoxins will irritate the gut. This will result in inflammation and tissue damage and cause immunosuppression, resulting in gut imbalance. Here I've got an image of a gel. Each of the lines on the gel represents the gut contents from one individual bird. Diet A and Diet B, you can see that the different lines on the gel, which represent different species of bacteria, are quite different between the different diets. And highlighted in the areas here, in Diet A, there's a species of bacteria which is hardly present in any of the birds that are given Diet B, and similar for Diet B compared with Diet A. So diet has a major impact on the different species that can be present, and any changes in constituents of the feed can put pressure on the gut ecosystem and also the gut balance. And under normal circumstances, this isn't too much of a problem and the birds will be able to cope with this. However, if there's underlying issues in the gut homeostasis, can be pushed that little bit too far and we can see upset. Water quality is another cause of intestinal stress. Water can be a source of bacterial challenge, but also the pH and the hardness of the water can influence physiological parameters in the gut. And here I've got an image of a drinker, which doesn't look very clean and is potentially a good or a large source of bacterial contamination. Management environmental stresses. During stress, certain hormones can be released, and these hormones can result in overgrowth of certain bacteria. In addition, heat stress can also cause gut damage. The development of the gut is also really important. If the development of the gut doesn't take place very well, this can lead to poor gut function, which will lead to malabsorption, and also a higher risk of dysbacteriosis and wet litter. Antimicrobials. Antimicrobials are used to treat enteric disease and bacterial overgrowth, but they also have an effect on the other members of the microbiota. So they don't just target pathogens, but they'll have an effect on many of the beneficial bacteria present there as well. So after antimicrobial therapy, the gut is left disturbed, and this will have an impact on subsequent gut function. The beneficial bacteria must be repopulated. So antibiotics slow the rate of development of the gut microbiota. Under normal circumstances, this process takes between 7 and 14 days. But in birds that have been administered antibiotics, it can take much longer. And in some cases, the mature microbiota never establishes. By inhibiting the gut microbiota, antibiotics can also have a negative impact, resulting in poor tissue development and shortened villi, poor gut integrity, impaired immune system development, which can increase susceptibility to disease. So extensive use of antibiotics will increase the risk of these negative effects. So having talked about all the different things that can have an impact on gut health, what can we do to reduce leaky gut? Well, maintaining gut health requires a strategic approach throughout the life of the bird. The birds should enter a shed that's clean and disinfected with sanitized water lines, and the chicks should be brooded correctly to help make sure that proper development of the gut tissues and the immune system takes place. We need to be able to recognize when the gut's likely to be challenged or stressed and be able to offer support to the birds at this time. Good biosecurity and disease control is really important, and the use of gut health products to aid gut during the life of the bird can help stabilize the gut following disruption, for example, after use of antibiotics. There are many different types of gut health products on the market, and listed here are some examples of those. Each of these different products has different properties, such as improving gut integrity, stimulating or providing beneficial flora, improving gut function, inhibiting pathogens, and reducing antibiotic usage. And it's important to choose the right product. It's also important to use the products practically and be sure that these are compatible with the different management practices available on the farm. It's important to remember that these products don't compensate for poor management factors, and poor brooding, water quality and feed quality will always have a major impact. These products won't necessarily give an improvement to FCR or yield, but they may reduce antibiotic usage, improve the litter quality, reduce foot pad lesions, and improve gut robustness. And here I just have a summary slide of all the different things that I've spoken about. I won't go and speak through them all, but I'll just put them up for you. So I'd just like to acknowledge my colleague, Dr. Richard Bailey, for all of his help and putting this presentation together. And also thank you all for listening.